Hello, everyone. Welcome to today's live broadcast, Unlocking Innovation in Life Science R&D, Overcoming Data Silos with Advanced Semantic Integration. I'm Bob Aliberta, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by LCGC and Spectroscopy and sponsored by LabVantage Biomax. And now, a quick word from our sponsor. LabVantage Biomax is a leading provider of cutting-edge solutions designed to empower organizations in the life sciences and pharmaceutical industries. Combining LabVantage's expertise in laboratory informatics with Biomax's deep understanding of semantic technology and knowledge management, LabVantage Biomax offers a comprehensive suite of innovative solutions, including the Ailani Semantic Search and Discovery Platform. By leveraging advanced technologies like AI, natural language processing, and semantic search, LabVantage Biomax enables organizations to accelerate R&D, streamline workflows, and foster innovation by harnessing the power of organized serendipity through interconnected data and insights. All right, we have a few important announcements before we begin today's event. First, you can submit questions for the speaker at any time during today's presentation by typing your questions in the Q&A box, which can be found at the bottom of the video player. You can also enlarge the slide window by clicking on the small icon in the bottom right corner of the media player, and those slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. It's time to get our event started, so let's meet our speaker. We're joined today by Dr. Verena Mertes. Dr. Mertes has a PhD in technical biochemistry and is Director of Project Management at LabVantage Biomax, with over 10 years of experience in life sciences digitalization. She has led key projects like protein production data integration and the development of a genetic engineering platform, and oversees Biotech 360. Thank you so much for joining us today. Dr. Mertes, please take it away.
Okay, yeah, so then welcome today for our webinar about unlocking innovation in life science R&D, overcoming data silos with advanced semantic integration. Yeah, so in this session, we will be exploring some of the key challenges faced by life science and biopharma R&D and how technology can help to address these issues, hurdles that researchers and companies face in advancing innovation. And from there, we'll discuss how semantic technologies, knowledge graph, and FAIR data principles are being applied to improve data management and integration, and how the topic generative AI is contributing to these efforts. We will also introduce Biotech 360, a platform designed to support the journey from early stage research through to product development. We hope that this session provides useful insights on how these tools and technologies can enhance your work in the life sciences. So let's get started. The challenges the biopharma industry is facing today are not new at all, but the real issue is that they have gotten even bigger. R&D costs are exploding like never before, and the pressure to innovate has never been higher. In 2022, the pharmaceutical industry invested $244 billion in R&D worldwide. 10 years earlier, that was only $137 billion. So this increase is remarkable. On average, it now costs about $2.3 billion to bring a new drug from discovery to market. This rise in costs is due to many factors, such as more complex clinical trial requirements, regulatory changes, and the challenges of operating in functional silos. At the same time as the cost to launch a new drug has increased, we have observed a sharp decline in peak sales over the past 10 years. Then, next we have the pressure to innovate. By 2030, for 190 drugs, including 69 blockbusters, patent protection will expire, and this will impact almost every major pharma company. So before 2030, in total, 236 billion of pharma sales are at risk. And as a result, there is a lot of pressure to develop new drugs and keep innovating to stay competitive. So the fact that these challenges are getting worse means that we need to act. So the key question for us at LabVantage is, how can we as LabVantage help biopharma companies to enhance their R&D productivity and boost innovation? We believe that the answer lies in two critical components: having good information and making good decisions. And these components are crucial at every stage of the R&D process, from planning and design to conducting experiments in the lab, analyzing data and reviewing results to decide the next steps. Every stage relies on diverse data sources, whether from in-house databases, public repositories, or specialized tools. It's essential that information flows seamlessly from one stage to the next. For this to happen, the right information must be delivered to the right people at the right time. This ensures that every step is data-driven and informed, empowering teams to make the right decisions. However, in reality, in many companies, the situation is far from this ideal. We often see significant challenges that disrupt the information flow and hinder innovation. There's dispersed data storage, for example, data on antibodies or samples or experimental results is often scattered across different documents, databases, and tools. And on top of that, internal data and public knowledge usually aren't linked, making it hard to bring everything together for a complete overview. There's also incomplete data sources, which hinders effective decision-making. The lack of metadata also makes it challenging to evaluate research reproducibility, which is crucial for validating findings. Also, without the necessary context, it becomes very difficult to generate meaningful scientific insights. Oftentimes, data are manually analyzed, which wastes time and resources that could be better spent on extracting insights. And last but not least, there's the non-compliance with the fair data principles. So when data isn't findable, accessible, interoperable, or reusable, it makes data discovery difficult or even impossible. All these challenges also make it very hard for executives to get the crucial information they need for key decisions. They slow down decision-making and can lead to incorrect conclusions. The struggle to find and use relevant information holds back innovation and productivity. When we look at the business impact, we see that organizations are losing a significant amount of time and money due to inefficiencies. For example, employees waste about 20% of their time, approximately 8 hours each week, on failed searches. Additionally, they spend around 15% of their time, or 6 hours each week, duplicating already existing information. And when you consider the broader picture, this amounts to a staggering 1.1 million wasted each year for every 100 employees in EU organizations. So this cost is significant, but we can't overlook the opportunity cost and hampered innovation, which can have an even greater impact, especially for biopharma companies. How to find data might cause you to miss chances to repurpose existing drugs, invent new therapies, and that can lead to lost revenue and missed market opportunities. It can also lead to overlooked safety signals that could potentially lead to clinical trial failures and, in the worst case, also harm patients. If critical information isn't accessible, discovering how drugs work can also be delayed, leading to missed opportunities to optimize the drug, failure to identify new indications, and it leads to extending development timelines. It also leads to inefficient collaboration, which wastes time and resources, reduces productivity, and delays your time to market. And there's a lot more what the impact is. So, as data becomes more complex, these issues will only grow. So what we want to do is to help you stop recreating the wheel and give your people time back for value-added activities like generating new insights. And how this can be achieved with our technology, this is what I will explain now. So first, I will explain how semantic technologies work, what they are, and why they are so essential for organizing and connecting complex data. Next, we will dive into the FAIR data principles, what they are, and why making data findable, accessible, interoperable, and reusable is critical for advancing research. We will then move on to real-world applications of knowledge graphs in life sciences, showing how they are being used to connect data in ways that delivers new insights. Lastly, we'll touch on the hot topic of combining knowledge graphs with generative AI, and how this combination is transforming the way we work with data and enhance decision-making in research and development. Let's begin with demystifying semantic technologies. What exactly are they and how do they work? If we look at the current discussions and applications of AI methods, semantic technologies frequently come up as they play a crucial role in addressing some of the challenges associated with generative AI. So let's dive deeper into what semantic technologies are and how they operate. And to do so, let's consider a simple example. If you have used an Apple Mac and have worked with files on your hard drive, you are likely familiar with how easy it is to search for image files. When you open Finder and look for images, it will quickly generate a list of all the image files on your drive, including PNGs, GIFs, JPEGs, and more. How the Mac knows which files are images? It's not something that many people think about or understand, so let's explore that a bit further. The Mac operates using an underlying ontology that defines various types of content, including file system items. This ontology helps classify not only the type of file, but also its content. For instance, image files like JPEGs, Photoshop files, and TIFFs are all categorized as images within this ontology. This ontology, known as the Universal Type Identifier or UTI ontology, is extensive and includes classifications for other file types as well, such as text documents, presentations, or archives. Ontologies standardize knowledge and understanding. They are structured frameworks or systems that formalize and categorize concepts within a particular domain. By providing a clear classification of different file types like image files, documents, and presentations, ontologies enable systems to understand and organize data effectively. So with an ontology like this in place, how does searching for content work now? From a user's perspective, if I want to find all the images on my hard drive, the system matches my query to a concept within the UTI ontology. Next, it identifies all the derived concepts associated with that search. And these concepts are then matched against an index built across all the files on my hard drive. And the result of this process is a Finder window that lists all the different images stored on my device. So essentially, this is a straightforward example of a semantic search system that many of us have been using for the past 20 years without even realizing it. So essentially, an ontology is a structured way of organizing knowledge. It defines abstract concepts like images and then specifies different types of images, such as JPEGs or Photoshop files. It also connects these abstract ideas to actual instances, so real files on your system that fall under these categories. And when you combine these abstract concepts and the real instances, which are also called individuals, into a network, then you create what is called a knowledge graph. So this network, made up of ontologies, individual entities, and the relationships between them, forms the foundation of a knowledge graph. Now, while the file type ontology I just mentioned only contains a few dozen concepts, imagine scaling that up for life sciences, where we could have hundreds, thousands, or even millions of interconnected concepts. With such a comprehensive knowledge representation, we can unlock powerful insights. And that's now where our Eilani platform comes into play. Eilani is our semantic integration and search platform, which leverages all those aspects: the power of semantic technology, ontologies, and knowledge graphs to integrate disparate public and proprietary data sources into a single unified semantic layer. On top of this semantic layer, we have built powerful applications like chatbots, analytical tools, and AI-driven insights provide a more effective way to analyze, explore, and leverage your data. A very central aspect of Eilani is that it makes your data fair. So what does that mean? FAIR stands for findable, accessible, interoperable, and reusable. 4 pillars that turn data into a valuable, actionable resource. The Pistoia Alliance has emphasized that one of the biggest hurdles to fully leveraging AI and machine learning is the sheer volume of data, but even more importantly, its quality. So how do semantic technologies, ontologies, and in particular Eilani help make your data fair? Let's consider an example of a pharmaceutical organization with teams working across different countries on an antibody-based therapy targeting HER2. Let's say clinical development is happening in Switzerland. Translational medicine is in China, and drug discovery is in the US. And here's a breakdown of their activities. In Switzerland, the clinical trials involve Herceptin, which targets HER2, commonly used to treat breast cancer. In China, the same target, now called ERBB2, is studied in a gastric cancer omics expression experiment. And in the US, Herceptin, which is the brand name, but which is also known as trastuzumab, is used in a binding assay with a CD340 receptor, another name for HER2. Now, even though all these groups are working on the same drug and the same drug target, they are using different names and synonyms, making it challenging to find connections. And this is where the FAIR data principle comes into play. By using semantic technologies, we can align these different synonyms into a single term. Now, all 3 synonyms for HER2 and the 2 synonyms for Herceptin can be understood as the same thing and aligned to a single term, thereby connecting the dots, setting the target HER2 and the drug Herceptin in the correct context, making information accessible to everyone. This example also demonstrates drug repositioning or repurposing. HER2 was originally identified as a target for breast cancer back in the late 80s, and Herceptin was approved for breast cancer treatment in 1998. Later, it was also approved for gastric cancer in 2006 and for esophageal cancer in 2010. So, the FAIR principles are all about improving the way we manage and share research data, and semantic technologies powered by ontologies are crucial for making that happen. Here on the left, we can see how assigning unique, persistent identifiers and detailed metadata help keep things findable. In the middle, through semantic mapping within the knowledge graph, we can combine data from different sources in a way that's usable by both humans and machines, making it interoperable. And on the right, by linking data semantically, we ensure it remains reusable for future research and computational analysis. Now, what you're seeing here is a screenshot from our Alani platform that lets us directly explore the knowledge graph and ontologies. You will notice the use of internationalized resource identifiers, also called IRIs, which is key to connecting data globally. Features like multilingual support, synonyms, and cross-references to other content sources are all baked into this system to enable the FAIR data principles. This semantic layer, powered by knowledge graphs and ontologies, organizes and connects all the data in a consistent and understandable way, making it accessible for both humans and machines. It allows us to build added value on top of the data, enabling applications like chatbots, advanced analytics, and semantic search. These features make it easier to interact with the data and extract more meaningful insights. And now I would like to show you some selected applications of the knowledge graphs. The first one highlights the use of the knowledge graphs for browsing or semantic searching. And then I will quickly demonstrate how knowledge graphs and machine learning, particularly generative AI, can be combined. So with a knowledge graph-based application, you can easily search for content by concepts. For example, you can drill down into specific domains like drug targets for a specific disease. Here you get a preview of the knowledge graph on the right side, which in this case is focused on the disease of interest. And moreover, you can further drill down your search result, focusing, for example, on available biologics relevant for the drug target of interest, which you see on the left side. Our Alani platform also offers an interaction trail, which records every step you take to zero in on the exact information you're looking for, which you can see here on top. This is a great feature for keeping track of your search journey. In addition to that, knowledge graphs make everything available for aggregated reports. For example, here's a gene report showing gene and protein isoforms, along with other details like associated diseases. The system also allows you to directly access sequence information and genomic context, displaying the gene features as they appear on their corresponding chromosome. It even lets you pull in additional related information, like 3D protein structures, which are linked to data resources such as PDB or AlphaFold. So, ontologies are not just about organizing concepts into hierarchies, as I have already shown. They also enable two key features that significantly boost the quality of your content. Essentially, ontologies act as a logical language that allows both for reasoning and validation. But what exactly does that mean? So let me explain. Let me give you a simple example using a wildlife ontology. Say we add a data point for a giraffe, noting that it eats twigs and leaves. From that information, the system can infer new knowledge, like the fact that a giraffe is a herbivore, even though that detail was not explicitly stated. Now, if someone adds data saying that a giraffe is also a carnivore, the system will flag it as invalid based on the rules built into the ontology. It knows that this can't be correct. So this ability to both infer new knowledge and validate content is really powerful and plays a big role in improving the overall quality of your data. As the last example, I would like to show you how to combine knowledge graphs and machine learning, and why this is relevant. Let me again give you an example. Let's say I want to find all the photographs in my library that show bursts. So today's machine learning models can easily do that. They can quickly identify bird photos. But as you can see here, they can also make mistakes, like identifying something as a bird just because it flies. So how do we avoid or reduce these false positives from machine learning models? And more importantly, is there a way to validate the output in the first place? So to achieve this, we have implemented a hybrid approach in our Alani platform that improves generative AI by combining machine learning with our semantic technologies. Here's an overview of how our platform Alani ingests content, builds knowledge graphs, automatically extends them, and makes them available for things like semantic searches, reporting, or even chatbots. And speaking of chat functionality, we have developed a hybrid AI approach that combines knowledge graphs with statistical methods like RAG, which stands for Retrieval Augmented Generations. And by blending these techniques, we greatly improve the quality of the answers. And most importantly, this combination helps prevent hallucinations. When you ask the system a question, the answer is grounded in the actual content resources integrated into the platform. So it won't make up answers or generate incorrect information. Now let me show you a quick example. You can put in a question: by which genetic factors is the severity of sickle cell anemia influenced? And Alani gives you precise answers and along with the supporting evidences. You can also go into details, for example, viewing the corresponding knowledge graphs and the related resources like diseases, or, for example, related protein sequences in UniProt. You can also keep the conversation going with follow-up questions. And most importantly, Alani only provides answers based on the content it has access to and nothing beyond what's integrated into the system. So, to sum up, Alani Insights enables private local large language models with improved accuracy. By combining RAGs with knowledge graphs, we deliver more accurate and informative responses. The system understands the context of your query better, providing more relevant answers. Everything is traceable. You can see the references and even drill down into the specific pieces of evidence that contributed to the answer. This leads to better results, especially for more complex questions, thanks to its reasoning and inference capabilities. Plus, the knowledge graph helps reduce the search space by narrowing down the content to what's most relevant. This means the system can handle large document pools, 60, 70 million full text documents or more, and still perform efficiently. So in the LabVantage ecosystem, we have got a powerful combination of capabilities. There's on the one hand the lab automation using the LIMS. Then we have AI and machine learning features that you might be familiar with through LabVantage Analytics. And when you pair these two with the search and integration capabilities of the Alani platform, we are perfectly positioned to tackle the challenges for life science and biopharma companies that we discussed at the very beginning. By combining all these capabilities through semantic integration, we are developing a new application to increase R&D productivity and innovation in life science and biopharma R&D. And this is our Biotech 360 initiative, to develop a platform to ensure that good information to make good decisions is available throughout the entire R&D process. We enable this by the unique combination of our technologies, the LIMS, our AI analytic capabilities, and the semantic technology, to deliver value at every step of the R&D process to increase productivity and innovation. With this initiative, we will empower organizations in life science and biopharma R&D to break free from information silos, to make data-driven decisions, to explore and find connections that were not obvious before, and to accelerate innovation and stay competitive. How we will do that? We at LabVantage Biomax, we will add our extensive expertise and capabilities that we collected in life science R&D over 20 years. We provide our fully configurable, flexible, and interoperable enterprise knowledge management system, which is based on the described proprietary semantic core technology, AI, and natural language processing. And it is also important that we don't have to start from scratch. Our product Alani for Life Science R&D will be the starting point for our platform that will be further extended. With Biotech 360, we are offering a pre-configured and pre-validated business logic tailored specifically for life science and biopharma R&D. It includes specialized knowledge bases, tools for managing and analyzing sequence data, as well as experimental data and advanced analytics. Biotech 360 integrates seamlessly with the LIMS and ELN systems and can pull in relevant structured and unstructured data from both public and in-house sources. And of course, additionally, it features the powerful semantic search capabilities combined with generative AI for enhanced insights. So Biotech 360 is built on a collection of research-focused knowledge bases, for example, for genomic information, plasmids, or antibodies that scientists can seamlessly navigate. They can easily select and use apps that are relevant to their specific research needs. And these knowledge bases are interconnected through the underlying knowledge graph so that researchers can gain new insights. For today, we will have a closer look at the plasmid app. Plasmids have a lot of applications in molecular biology and biotechnology, for example, to develop new vaccines or gene therapies. Our plasmid knowledge base is a unified database for storing, managing, retrieving, and analyzing plasmid sequences and related data in a fair way. Researchers can easily access and share plasmids and related information. And it integrates with bioinformatic tools, public databases, and enables to trigger, for example, LIMS workflows and feedback experimental results. So let's go to the web portal of Biotech 360. Here you see the different apps I was talking about. For example, the app for genomes and plasmids, nanobodies, or different apps for drugs and drug targets. Now let's have a closer look at the plasmid app. So in the central section, you see the repositories of all the plasmids uploaded to the system. And each plasmid comes with a mini report that shows the most important information, like description, plasmid size, encoding, gene insert, and it also comes with a plasmid map. This map is interactive, so you can, for example, zoom in and zoom out. You can also display or undisplay features, and you can also toggle between circular and linear view. You can also edit plasmid information. For example, here we have configured editable plasmid reports, for example, where you can edit the expression system, or for example, the vector backbone, and so on. You can also bookmark or tag your plasmids so that you can easily access them later, organize and classify them, and also share them with your colleagues. And of course, you can select the plasmids and export them in different formats, for example, in sequence formats like GenBank or FASTA. On the left-hand side, you can see different filters where you can filter plasmids according to their name, according to who has created the plasmids, according to vector backbone, vector type, but also according to plasmid sites. Also, you can filter plasmids by insert information, like the gene that is inserted into the plasmids, its most similar UniProt entry, Or you can filter plasmids by the combination of certain DNA parts, like combination of promoter or together with a certain coding sequence, and you can filter by cloning and expression system. Now let's look for all plasmids which have the EGFR gene as the inserts. So when we do that, we see 3 plasmids as a result in the middle. And now let's have a closer look at the first one. So on the left-hand side, we have more detailed plasmid information. And in the middle section, we see all the plasmid features and get direct access to the feature sequence. We see more information about the gene that is inserted and, for example, the associated UniProt entry and the associated gene in the human genome. We see also plasmids encoding similar proteins, and we can access the plasmid genealogy. Under experimental data, I can also trigger a LIMS request. Let's say, for example, I want to request the amplification and purification of the plasmids. Then I can enter the relevant information for the lab technicians. So let's make that a bit faster. And then the request can be submitted to the LIMS. Under pending requests, you see all the requests that have been submitted, and then under completed requests, you can check all the requests that are completed within the LIMS and the respective results. Okay, so everything you just saw is represented in the underlying knowledge graph. And here's a basic snapshot of the knowledge graph. We start by cloning DNA into a plasmid. The plasmid produces a biologic, like a vaccine. The biologic targets a specific gene in the human genome, which we can find in the RefSeq database. The gene might be linked to a disease with references available in PubMed. The plasmid can also be transformed into a strain that expresses the biologic. And the strain can then be used in fermentations to produce the biologic, which is then extracted, purified, and tested for its biological activity. Finally, R&D reports are created to document the findings and results from these experiments. So with our knowledge graph, we can cover the relevant building blocks for life science R&D, including, for example, molecular biology, experimental and process development data, public knowledge, and any internal documentation that is generated along the way, connected and make it easily accessible to generate new insights. To sum up, Biotech 360 will provide multiple benefits, not only for researchers, but also for production teams and organization leaders. With Biotech 360, you can transform your organization's decision-making process from discovery to production. You can connect to LIMS to trigger lab workflows and enrich your knowledge base seamlessly. It accelerates R&D by fostering better collaboration and achieving faster, more efficient research outcomes. Plus, it enables you to optimize resource utilization and minimize waste. Biotech 360 is also designed to adapt and scale effortlessly, growing alongside your organization's evolving needs. And by ensuring fair data, you will benefit from greater transparency, reproducibility, and support for AI-driven innovation. To ensure Biotech 360 closely meets industry R&D needs, we collaborate with industry partners in its development and invite you as well to join us so that you will benefit from some key advantages. You will get early access to the platform, giving you a head start and using cutting-edge tools to gain a competitive advantage. You will have the opportunity to influence its development, offering direct feedback to ensure Biotech 360 is perfectly aligned with your R&D needs. We provide ongoing professional support and enhancements so you can trust that the platform will evolve and improve with time. Plus, as Biotech 360 grows, you will benefit from future product developments while keeping configurations tailored specifically to your unique needs. And now I would like to come to an end, and we hope you found this session valuable, and we look forward to explore new opportunities together.
All right, thank you for such an informative presentation, Dr. Mertes. Before we get started on the question and answer session, I would like to quickly remind our audience how to submit questions. You can submit your questions by typing them in that Q&A box, which can be found directly below the video player. So let's get started and jump right in. Our first question of the day: what industries outside of life sciences can benefit from these technologies? And do you have specific case studies?
So yes, our technology is actually industry agnostic, and it can be applied across various sectors. We have developed specialized versions of Alani for industries like chemistry, materials science, or food and nutrition. So in chemistry, for example, Alani comes with a knowledge base of over 10 million compounds and reactions from various trusted resources. And it provides chem informatic capabilities like optical structure recognition or advanced chemical structure and substructure searches. In material sciences, for example, we help to identify sustainable materials by connecting diverse scientific data. And in food and nutrition, we can, for example, help to streamline product development by integrating data on nutritional content, health outcome, and other relevant sources.
Thank you for that answer, Dr. Mertes. And our next question from the audience: how does the Alani platform handle large data sets across multiple sources? And how scalable is it?
Yeah, the Alani platform is designed to handle large, complex data sets from multiple sources. It can efficiently manage millions of documents and large volumes of structured and unstructured data. And Alani uses the knowledge graph to narrow down the search space and retrieve only relevant information, making it scalable without sacrificing performance or precision.
Well said. And moving right along, our next question from the audience today: what are the typical integration challenges when implementing knowledge graphs in existing systems? And how does your platform address them?
Yeah, one of the main challenges we have observed when integrating knowledge graphs in existing systems is the growing complexity that comes from handling multiple data sources and their interrelationships. Agile solution building process where we address this by breaking down the complexity into manageable tasks. So each data source is connected to the semantic model in a step-by-step approach, making the overall integration more structured and easier to manage, while we still maintain the richness of the data connections.
All right, great info there. Our next question: how do you ensure data security and privacy when dealing with sensitive information, especially in industries like biopharma?
We ensure data security and privacy by leveraging the semantic model, which allows us to set granular access controls. This means we can assign different privilege levels to each attribute, and ensuring thereby that sensitive information is protected while still enabling secure access to the data that's needed.
All right, looks like we'll wrap up with this question: how does the system validate data and prevent incorrect inferences from being drawn, especially in complex data sets? Can you speak to that a little bit?
Yeah, the key to preventing incorrect inferences lies in maintaining semantic integrity, particularly within the ontology. The system performs inference checks to ensure consistency and identifies any logical contradictions in the data. And this allows the platform to validate new information against the established structure of the knowledge graph, flagging any issues and ensuring that only accurate, consistent data is used, even in complex data sets.
All right, with that, we will wrap up today's event. I want to thank the audience for attending and for participating in the Q&A. I would also like to thank our sponsor, LabVantage Biomax, for making today's educational webcast possible. We'd like to ask everyone in the audience to participate in a brief survey. This survey will appear on your screen after today's presentation has concluded. You'll also receive an email alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks to all for joining. We'll see you next time. Goodbye, everybody.